So today we're going to be talking about the 5 agents of evolutionary change, and I do have my notes down here to help me. Mutation is first. So although mutation is the original source of all genetic variation, mutation rate for most organisms is pretty low. So the impact of brand new mutations on allele frequencies from one generation to the next is usually not large. Then we have non-random mating. In non-random mating, organisms may prefer to mate with others of the same genotype or of different genotypes. Non-random mating won't make allele frequencies in the population change by itself, though it can alter genotype frequencies. This keeps the population from being in Hardy-Weinberg equilibrium, but it's debatable whether it counts as evolution since the allele frequencies are staying the same. We have gene flow, and gene flow involves the movement of genes in or out of a population due to either the movement of individual organisms or their gametes. Organisms and gametes that enter a population may have new alleles or may bring in existing alleles, but in different proportions that those already in the population. Gene flow can be a strong agent of evolution. Then we have non-infinite population size or genetic drift. Genetic drift involves changes in the allele frequency due to the chance events. It's literally like saying a sampling error or a sample error in selecting alleles for the next generation. Drift can occur in any population of non-infinite size, but it has a stronger effect on small populations. Then we also have, of course, the last one, natural selection, which everyone knows. A natural selection occurs when one allele or a combination of alleles of different genes make an organism more or less fit, that is, able to survive and reproduce in a given environment. If an allele reduces fitness, its frequency will tend to drop from one generation to the next. We'll look into details at different forms of natural selection. So natural selection has 3 types, selection type. They have disruptive, directional, and stabilizing. So the first one, the stabilizing selection, in stabilizing selection, intermediate phenotypes are more fit than extremes. So for an example, if you get a frog, so you get a green frog, and they might be best camouflaged and survive best on a forest floor covered by medium green plants. So stabilizing selection tends to narrow down the curve as well. Directional selection is one extreme phenotype is more fit than all the other phenotypes. So for example, like you have a green frog and a brown frog, and they move into a new environment, and it's with dark soil and vegetation. So the green frog might be less hidden than the brown one, and survive better. So the directional selection shifts from the curve towards the favorable phenotype. And then you have disruptive selection, where both extreme phenotypes are more fit than those in the middle. So example, if those frogs move into a new environment with patches of light green moss and dark green shrubs, both the green and the brown frogs might be better hidden and survive better than the ones that are like kind of like in between the green and brown. This diversifying selection makes multiple peaks in the curve. So then we also have the concept of species and speciation. So the species concept defines as species as members of population that actually or potentially interbreed in nature, not according to similarity of appearance. Then the concept of speciation is how a new kind of plant or animal species is created. Speciation occurs when a group within a species separates from other members of its species and develops its own unique characteristics. Thank you.